I'd like to welcome you to Teledyne CETAC Technologies webinar. Discover a new choice in ICP-MS low-volume sample handling, the MVX-7100 microliter workstation. I'm your host, Bob Armbruster. Today, our automation product manager, Brian Cook, will be giving a presentation covering the different aspects and capabilities of the MVX-7100 microliter workstation. I'd ask that you please save any questions till the end of the presentation when Brian will actually answer the questions then. Just a note that the webinar will be available for download on our website in the next few days. On that note, let's get started. Brian, please start your presentation.
Thanks, Bob. Thank you, everyone, for joining us. Today's webinar will be on the MVX-7100 microliter workstation and how this automation system is able to change your ICP-MS. We'll start with an overview of the MVX-7100. For those of you who are unfamiliar with this system, this is just a quick picture of this, and you'll see that it includes our automation system along with a full-height syringe, our half-height syringe, and our valve system. And that is a 384-well microtiter plate in the background. So a quick overview of the MVX-7100 automation microliter workstation system. What this system is able to do is it's able to reduce reagent and sample volume usage by using well plates. We're able to do some sample mixing, and we'll also discuss internal standard addition, septum piercing, and what the syringe-driven performance characteristics we're able to gain with this system, and how we're able to do advanced sample control. First question is, how did we get to this point? The problem that we were having, customers in the field were coming to us and saying, what can I do? I have a small sample size with elements that are already at trace levels. These customers, typically the solution is dilution, but obviously this causes a loss of sensitivity. These customers are already working with ICP-MS, and they're having to look at multi-collector or high-res systems to try and gain that sensitivity. So we're trying to find another option. Another question is low-flow nebulization products for self-aspirating. Really takes a lot of practice to get your matrix matched so that you have a consistent performance. And then finally, the automation. Just standard automation isn't meant for low-volume applications, and the ones that are out there are meant for HPLC or GC industries where they're really not worried about non-metallic components. So it was really just developing something specifically for the ICP-MS industry. This isn't the first time this question has been asked. In a review paper by Todoli and Mermet in 2006, and as well as other papers, they discussed several different sample types that they came across. And you can see on our list here we have cells in the sample volume of 20 microliters, nanoparticles, brain at 1 milligram, and metalloproteins at 50 microliters. And they're starting to see a lot of fields in which the available sample volume is a limiting factor for elemental analysis. So the first question we usually get is, how does the MVX-7100 microliter workstation system work? So what we have here is a quick diagram to try and show you how this system operates. We have the MVX-7100 microliter workstation on the right, in the middle our valve syringe model, and on the left our syringe pump module. The syringe pump module works to feed the ICP-MS. So it is feeding, the blue line represents the rinse or carrier solution that we're pushing into the ICP-MS. While that is running, we have the MVX-7100 workstation go out to the sample, pierce the septum if needed, and then begin the sample uptake. This is controlled by the half-height syringe on the valve syringe module that is below the 6-port valve, as indicated by the green flow pathway. So we use that syringe to pull up the sample volume into the sample loop. Then we move to the inject phase. At this point, we put the sample loop in the pathway of the syringe pump module, which then injects that into your ICP-MS system. Meanwhile, we use the half-height syringe to rinse out the internal portion of the probe in the rinse station, as indicated by the blue pathway. So the MVX-7100 automation portion, I'm going to break these components down one piece at a time. What really separates this automation platform is we're able to run 96 and 384 well plates. At the same time, we're able to do septum piercing, and this is a 2-tray format. What's really unique about the MVX-7100 system is that we offer stepper motor control for the X, Y, and Z axis, along with encoder feedback for precise precision control. And why this is really valuable is a 384 well microtiter plate has a well opening of 6.2 millimeters. So we have to ensure that we always know where our system is at if we want to have the accuracy to be able to hit these small microtiter plates. Also, we designed this to be modular. And what this allows us to do is we're able to move the which is the portion that you typically would want to have close to your ICP-MS so that you can be set up for a right or left-handed instrument. We've also allowed this to be scalable. You're able to add additional syringe modules should you want to do more advanced operations with this. The automation portion also has a dual flowing rinse with internal syringe rinse control, which I referenced earlier, using that half-height syringe to rinse out the internal portion of the needle while you're washing the external portion, using your 2 different flowing rinses. The SPM-700 Plus module. This is included with the MVX-7100 base system. It is a full-height syringe. And how I usually explain this is this is the peristaltic pump replacement. This is what you use instead of that peristaltic pump to give those smooth, stable baselines, even at low flow rates. We're able to do flow rates less than 5 microliter a minute, but we're not limited to low flow applications. Depending on the syringe module, we can the syringe barrel you have selected, we can even have flow rates exceeding 1 milliliter per minute. And you're able to have multiple SPM modules connected to one MVX system. Our system ships with one 2.5 milliliter barrel for the SPM-700 module. The second part of this is going to be the VSM-700 module. What's unique about this is it's a flow injection valve. Now this is really one of the starts of the separation. It's a flow injection valve for low volume, and we use inert materials. We're using a PFA rotor and a PFA stator and a peak rotor. It's a 6-port valve. Also, one thing we've done is we've included an additional loop between the 6-port valve and the syringe so that we ensure that we never pull actual sample into that borosilicate glass syringe to cut down on any carryover. We have the half-height syringe on bottom for our precision Aliquot Control, which we're able to do aliquots down to 5 microliter all the way up to and greater than 1 milliliter. This allows us to do sandwich loop loading, which we'll discuss later. And this system ships with a 1.25 milliliter barrel. As a side note, we have different barrels for both the half-height syringe and the full-height syringe, depending on what application you're looking at. The MVX-7100 software. We have a standalone software package, which is very flexible. We communicate via instrument triggering with the ICP-MS, so it's compatible across a wide range of instruments. In our software, you're able to select full or partial loop load, along with in-vial reagent addition. What's really great about this is it allows for complete control of the components individually, along with sophisticated rinse controls to really maximize what you can get out of this automation system. One lab that we've worked with, we were able to really show some great improvements in efficiency for them. So before, they were using 100 microliters of sample and 4.9 milliliters of diluent And at that capacity, they are running about 80 samples in 8 hours. After switching over to our MVX-7100 microliter workstation, they were able to cut down their sample to 20 microliters and only 0.9 milliliters of diluent. At the same time, they were able to increase their sample capacity to 192 samples in 5.5 hours. So we're able to use 5 times less sample and reagent volume, while at the same time increasing their capacity by greater than 2 times and reducing that time by 2.5 hours, along with reduced autosampler footprint. This was really kind of a step change for them in how they're handling their ICP-MS automation. The supported racks that we have in our system, the first one we have is the 48-position 1.5 mil vials. Vial rack system, which is in the upper left-hand corner. These are PFA vials, and this is adapted from our popular ASX 110, 112 automation system, where people really need the PFA option. Second, moving clockwise, would be the VT54, which is the black rack with the 2 mil polypropylene vials. There's 54 positions per rack. And this is the rack you're probably most used to seeing on probably a traditional HPLC style system, if you've seen some of those around in your lab, or possibly a GC automation system that you may use. We're also able to be compatible with 96 well plates, which have variable volume up to about 1.5 milliliter per well, and the 384 well plates, which I've seen those up to 400 microliter per well. So you can really see that you're able to really customize this to go with the workflow that you're using in other portions of your lab. The next portion I want to talk about is the tempering module. Now this is the first optional accessory that we're talking about. We have the same rack compatibility for the 48-position PFA, your VT54, your 96 and 384 well plate. We're able to temper between 5 degrees C and 40 degrees C using this Peltier module. It's especially useful for volatile samples or metal tag proteins, keeping them in their natural state as you want to analyze them. And as I did stress before, this is an optional accessory. You'll notice the 3 LED format we have on here allows you to get a visual feedback just by looking at it as to where we're actually at related to the set temperature. Red would be heating, and green when it becomes steady at the correct temperature. So what really separates this from other low-volume automation systems that you'll see on the front end of your HPLC or GC style systems is that we have a non-metallic flow path. This really took a lot of research on our end to find something that was able to work in the same capacity while being non-metallic. So we use a quartz needle along with PFA tubing, and then we have a CTFE 6-port valve along with a peak rotor, and then a CTFE syringe valve for the top of our syringe. And then we use the borosilicate glass for the syringe. And as I stated before, we keep the sample solution from getting into the syringe or into the actual barrel syringe so that we can ensure we have the best possible performance. Some of the other features that really help separate this from other products are syringe-driven rinse for more stable self-aspiration across different matrices. This system allows you to maximize your data, both integrity and quantity of data, with limited sample volumes. Segmented sample loading, which we'll talk about later, has really been one of the key features that outside of the ICP-MS industry is a fairly common technique. But here in the ICP-MS industry, this is kind of a new concept for some people. You're able to also use syringe-driven internal standard control. And then we can do septum piercing for sensitive samples along with our temperature control option. Just a quick overview of some of the sample performance. You can see at 100 microliter sample volume, we're able to get a less than 1% RSD, and this is without internal standard correction. And from another lab that has used this, showing less than 0.01% carryover on our system. So for your internal standard edition, you have a couple different options. Some people choose to add an additional full-height syringe module so they can have that keyed in after to give the best control for their internal standard. Some customers will still want to use the diluent to add their internal standard. And then you could also use the ICP-MS peristaltic pump for your internal standard edition. It's just you're going to lose some of what you gain by using the syringe if you're using the peri pump for your internal standard edition. Now, I've talked about septum piercing a couple times. I've mentioned that. And I really want to talk about this. We have a couple different options when it comes to septum piercing. We're not just talking the Teflon film, the 2 mil Teflon film that you see below here. Yes, of course, we can pierce that Teflon film, but we're also able to pierce your traditional silicone septa with a PTFE lining. And we have septum piercing options for the 2 mil vial, 96 and 384 well plates. Sample handling. Now, you've heard me mention a few different times talking about advanced sample loading. So the question I usually get from people is, what is this advanced sample loading? Well, this is kind of a quick overview. So we took a sample loop and we're showing this as a linear representation. So the first one is going to be your full loop, which is what you'll see on a lot of flow injection systems out on the market, including our AS Express Plus system. Is what you're doing is you're pulling volume that exceeds the capacity of your sample loop. So if this was in the valve, you would have sample on both sides of the sample loop. Now this is great, it gives good performance, but the problem that you run into there is you do have some wasted sample because you're pulling it through on both ends. So the next one below that, you'll see a sample and air. And what we have here in the middle is we have our sample. And then circle on either side, you'll see we actually have an air bubble on either side of this. So that is a discrete aliquot volume that we're actually using, which is no sample waste, which is the most common way people are utilizing the system. Another injection method that people have been using with this is sample and reagent. So that's the one we have represented on the bottom here. So we have an air gap followed by sample, then an air gap, and then a reagent with another air gap. So there we're actually doing two discrete aliquots inside that same sample loop. Why do you want to do these? How do you select which type of sample handling you want to use? So the full loop injection is probably what most people are common with from the flow injection systems. So again, you're overfilling the loop. Typically, you're able to get a little better speed performance with that, but you are going to waste some sample. Below, you'll see some data we have from one of our posters where we look at a 5 microliter sample loop installed and doing a full and a partial loop on a couple different elements. And you'll see what kind of performance, and that's a 5 microliter sample we're talking about there. Even a partial injection, we're at 2.4% RSD on lead. And then right below that, we have a 20 microliter loop. So we're doing a full and then a partial loop injection to show what sort of reproducibility you're able to get with that. Probably the largest one that we're seeing is sample with air. And the reason for that, the big things that people are stressing is there's no sample being wasted with this. We are injecting every drop of the sample that we pull up into the system. It really maximizes what you're able to get out of that. And also you're able to get improved washout. So what you typically see with trailing on ICP-MS is you're always going to have some spreading of the sample into the rinse or carrier solution that's pushing it. With this small, can be 5, 10 microliter air slug, it's not going to disrupt your ICP-MS plasma, and you're able to really get kind of a squeegee effect that helps push out that sample, and you don't have that spreading into your rinse and creating those longer washouts. And in some other situations, we've actually had people use a sample with reagent. So sometimes they're doing in-loop chemistry. So if I go back, you'll see on this one we actually have an air slug between the reagent and the sample. We have some customers that are choosing to not have that air gap. They're trying to really introduce some in-loop chemistry. Other people are using that to help clean out lines. Maybe they're having some sample and they want to throw a Triton X solution in there right behind so it cleans right as it goes into their system. There's really a lot of different things that you can do with this. The system ships with 6 reagent vial positions that you're able to utilize with that system as it ships. So with this Aliquot Control, and I'm talking about doing our partial loop injection here, where we're doing sample and air. How precise is this really? So we did some testing with this, and this graph is a representation of variable volume taking from 1 10 microgram per liter solution. So what we have here is we have a blank solution, and then we took a 25, 50, 100, and 250 microliter sample in the sample loop and injected this. So what we did is we did a calibration using areas compared to height. Then what we did is we then ran a solution of full 250 microliters at 1 microgram, 5 microgram, and 10 microgram per liter so that we can correlate the area under the curve and show that even if you're not changing the aliquot volume in the sample, it will still give you the correct concentration. And you can see our calibration factor was 4 nines, and our readbacks on cobalt 1.04, 5.06, and 9.95. And once again, that is calibrating on a 10 microgram per liter solution using variable volume and area underneath, and then reading back a full 250 microliter slug to show that the area under the curve does correlate. Now on this, we're also using rinse for makeup volume so that we don't interject for a 25 microliter. We wouldn't want to inject a 200 microliter air slug with that. So we use rinse solution to make up that difference so that we don't disrupt your ICP-MS plasma. What are some of the applications that we've seen on this? We'll discuss a few of these, but isotopic ratio, zinc and protein isolates, cisplatin and cell lysates, some whole blood applications, along with nanoparticles and micro drilling. So we'll start here, isotopic ratio. So with this, people were looking at doing a steady state signal. So one of the things they really found valuable with the MVX-7100 system is that syringe-driven rinse works across varied matrices. So if they're getting a couple different matrices type, but they really want to see how that isotopic ratio changes between the two, they don't have to worry about using a self-aspirating neb and trying to get the same performance from each one of those. The example below is using only a 20 microliter sample with our DS5 total consumption nebulizer. Another application we have had is zinc and protein isolates. I'll probably butcher this, the Xenopus laevius, South African clawed frog. This is an example where they used 50 microliter sample before digest, and this kind of shows some of the spike recoveries and standard materials and what kind of performance they were able to get with this system. Really allowed them to push to lower levels and be able to use that same sample across multiple types of analysis. Cisplatin, I'm sure many people are familiar with this, used for cancer treatment. And what they liked about our MVX system is they were able to improve washout with our flow injection valve system and able to use less than 100 microliters of sample. Nanoparticles is something you're really starting to see a lot of this in the news, and we've done some with silver nanoparticles at different solutions. Hold on, a little technical issue there. Silver at different dilutions, and again, they're using a steady state signal, and they're looking for the size distribution of those materials in that cell culture. So optimization for microsamples for ICP-MS. So this particular application, we're looking at combining discrete aliquot sampling. So again, that's taking a well-defined sample volume and using that with our total consumption nebulizer, our DS5. In this case, we have a flow rate of about 8 microliters a minute. So you can see at these low flow rates, a 20 microliter injection with our total consumption nebulizer, you're able to see what sort of data we're actually able to generate. And that's usually only 20 microliters on the left-hand side there. And even if we go up to 100 microliter injection using a standard nebulizer and spray chamber, you're able to see we're able to actually generate some really good data using a very small volume. And again, that's the other part of this, because a part of it is using a small volume. But people that are using these small volumes are typically also the same people that really need to have the highest data integrity. They need to get the best performance they can from this. And that's why we were using the syringe-driven system for these applications. So you're probably wondering, how does your data look when you're using this? So up here we have a chart showing repeatability of 7 replicates with our MVX-7100 using our DS5 total consumption nebulizer, which flows around that 8 microliter a minute. You're able to see we're able to get really good RSDs on this. Most of these are below 1% RSD while running at these really low flow rates. So that's something I always try and stress with the customers when we're talking about this, is that just because you have small volume, it doesn't mean you have to give up on getting the best possible data. This is really about getting the best data while minimizing your usage of sample. Another area of interest is geochemistry. So one of our partners, they had limited sample volume, so they're working with, I'll go with the unscientific name of snail shell, and they're studying the different elements across time to find out how this reflects the environment in which this was raised. So with this, they were able to not have to waste sample. They're starting out with only 100 micrograms of powder. So they're drilling out and trying to get a profile for as these animals grow, how do they accumulate this in their shell. Another one with this is we're able to also pair this with our Aridus 2 system to help even drive the performance even lower on the systems. And you can see some of the data. They can show how iron increased as this aged. Another area that we've seen a lot of interest is in clinical applications, in blood applications specifically. Whole blood is a difficult matrix to work with. One of our customers is usually just using this for lead, mercury, and arsenic in blood. So what they loved about this is they're able to increase their throughput while at the same time reducing their sample and reagent. One thing I like to stress with this application is we've shown a lot of these where we're running at this 50 microliters per minute. But that doesn't mean that's what you have to use that with. This customer was using this at 400 microliters a minute, a much more standard ICP-MS flow rate. Also, we've had people do research with titanium used in implants and prosthetics and how that flows through your system. Several other applications where we've been getting a lot of interest in the clinical industry is, of course, blood, which we've talked about a few different applications there. Also urine, testing for multiple metals there, and biological samples. Again, anything where there's limited volumes. We've had people come and talk to us about serums. Anything you can think of where there's only a limited amount. In the geological industry, of course, shellfish and the micro drilling. And any other precious sample, when they're trying to date stuff, they're trying to go back and look at a historical reference. Very often we don't want to consume this entire sample for one analysis. you're not going to be able to get the permission to use that if you're going to consume it. But we're able to take small volumes and use this. So not dissimilar to what some people have done with laser ablation systems that we use, by just taking a small laser shot and being able to use just a small sample to get data. This is another take on that. We've seen a lot of different interest across academic research, across multiple disciplines. We have people in analytical research that have reached out to us, also environmental, geology, medical, and toxicology. Really, there's a lot of possibilities for what the system can be used for. In industry, we're talking with people in fuels industry, because obviously you're using volatile elements, so if we're able to introduce those at lower flow rates so we can do less dilution and get better performance. In the nuclear and the semiconductor industry, again, they're looking typically at very low detection limits with small sample volumes, because those can be very dangerous to work with if they're radioactive, or it just can be a rare earth element where it's hard to get a lot of those. So you just have to really get as much value as you can get out of those sample types. Well, I put up here a couple different resources. We have our MVX-7100 brochure, along with a link to some applications notes. And then any sales questions, you can link there and find out who's going to be the person to contact in your area. And I also have my email. I'm going to attempt to share a video with you guys, 1 second, showing our MVX-7100 system in action. Just 1 second as I get this set up. And what I'm going to show here is this is our MVX-7100 microliter workstation. And if you can't see it on here, you'll be able to go to our website and see this video. But I'll pause it at the end. And what we're showing here is our ability to run that 384-well microtiter plate and showcase our 2 syringe-driven system along with that 6-port valve. So this is a time-lapse video of us showing the precision and accuracy, and then also showing the sample mixing in these small plates. You can see we took a yellow and a blue dye, and you can see it mixes in there pretty well to give that the green color. And then we let that dry overnight. So I'll pause that here so we can see that. So you can see this is just something we threw together to just kind of help demonstrate. And again, each one of those is about 6.2 millimeters opening on that 384-well microtiter plate. 1 second as I switch back to the PowerPoint here. With that, we'll go ahead and open this up to any questions that we have coming in. All right. Looks like I got the first question here. We had someone ask, what comes with the base system? So when you order the MVX-7100 system, you will get 2 384-well plates, polypropylene, 2 polypropylene 96-well plates, 2 reusable VT54 racks. Along with that, we'll provide 200 2-mil polypropylene vials, 2 96-well plate Teflon films, 6 20-mil reagent vials, and then, of course, you get the MVX-7100 Automation system, along with the SPM-700 full-height syringe with the 2.5-mil syringe barrel, and the VSM-700 valve syringe module with a 1.25 milliliter syringe barrel, and all tubing that you would need to plumb the system. So if you have the ICP-MS, you should be able to order this MVX-7100 system and be out of the box with the install. We'll come and set that up for you. The system does require an install, and that comes with about 8 to 12 hours of installation and training time, along with a little bit of method development to get that set up and organized for you guys. All right, I got a few more questions coming in here. First one, how much is the system? Again, for that, I would reach out to your local sales representative. They would be able to provide you any pricing depending on what options you may need. I have another question here is, can it handle crude oil samples with xylene matrix? We're working on the full chemical compatibility list right now. I believe xylene is listed, but I will verify if we have xylene as a listed compatible material, and I can get that posted back to you. Do we have any other questions coming in? If you think of some questions after the webinar, please feel free to send me an email or give me a call, and we can talk about what options and questions that you have. We have just another one coming on. Is boron, if boron is an issue, are there alternative syringe barrels? So as of right now, we only have the borosilicate glass option for the syringe barrels, but we are working on getting another option for you. Will there be an HF compatible kit? That is something we are working on at this time. We don't have one to release at this moment, but that kind of ties in with the alternative syringe barrels. We've been really trying to find an HF compatible kit to get fully inert materials. And I have a question here on DMSO. DMSO should be compatible, not on the rinse station, because the rinse station for the MVX-7100, we use the Perry pump, but I am showing it as compatible. Full compatibility list here available in a couple weeks. What is the lowest sample volume it can handle? We state we can go down to 5 microliters. We've done actually testing down to 0.5 microliters. For best performance, I would say 5 microliters and up, but certainly there are plenty of options we can look at with different syringes to try and maximize what we get out of the sample. The next question I have is the system being utilized for trace metal analysis for 200.8 or 60/20 analysis? And currently we don't have anyone utilizing it for those analysis at this moment. Most of those customers that I've talked to don't have a need for smaller volume, but I think there's probably a few applications where people are running it where they do have some limited volume applications. Certainly it'd be something we'd be willing to try and put together, do an application note possibly, or work with a partner to try and get some of that data. There's a lot of really good questions. Please keep them coming. Trying to see if I missed any questions here. Hold on as I read through and just verify if I've missed them. I think I got all the questions answered here. Like I stated before, if you do have any additional questions, please feel free to email me or your local sales member, and we can get back to you with the compatibility. As I stated, I hope to have a full compatibility list here in a couple weeks that we can have posted on our website to really help simplify this process for everyone. If there are no more questions, I would like to thank everyone for your time, and thanks again for joining us.
Have a great day.